friends, I just wanted to make a quick video where I help you make the mitosis note cards that we made in class. So hopefully at this point you've at least made part of them and you're just looking for specific ones. So feel free to scroll through the video and pick up wherever you need to finish the note cards. I will be doing them from the very beginning. So starting with the first note card. On side 1, we're just going to write mitosis. And on the other side, we're going to define mitosis. So mitosis, what is it? Well, mitosis is a process of cell division in which a single cell divides to form two identical daughter cells. Okay, now this is important that it makes two identical daughter cells. You'll learn later about another process of cell division that makes different cells. Oh, sorry, I did not realize I wasn't centered. So there you are. So you'll learn about the other type of cell division later at some point, but for now, mitosis makes two identical daughter cells. And mitosis includes prophase, metaphase, anaphase, and telophase. Telophase, otherwise known as PMAT. Notice it does not include interphase and it does not include cytokinesis. So go ahead and pause the video here if you need to write this down. I am going to be moving on. Okay, so moving on to our next one, we're starting with interphase. So on this front side of the note card, I wanted you to label interphase at the bottom. And then now dry cell and interphase. So we're drawing cell membrane with a nucleus with a lot of genetic material on the inside. Notice that it's strong. It's long and stringy. This is chromatin. And then I'll go ahead and label my nucleus and cell membrane. So this is what a general cell looks like when it is in interphase. The DNA is long and stringy. It has not been condensed yet to form a chromosome. Now let's write what's happening in interphase on the back. Okay. So now on the back, we're saying that interphase is when the cell grows and develops. DNA is in chromatin form. Meaning that it's long and stringy. This is where the DNA is copied. Because remember, if we want to have two identical daughter cells at the end of cell division, at the end of mitosis, then we have to copy the information that we're going to be passing on before we can actually do the dividing. Again, interphase is not part of mitosis. It happens before. And the cell is in interphase about 90% of the time. So go ahead and get all of that copied down, and then we'll be moving on to prophase. Okay, so prophase. Here, give yourself some space to draw. We're going to be drawing the cell and then off to the side a chromosome, a close-up of it. So prophase. Okay, so we have our cell. We have our nuclear membrane or nuclear envelope. It's starting to break down though, so I'm going to draw it as a dotted line. And then draw some little chromosomes on the inside. Note, I only drew 3, but a human cell has 46 chromosomes. Then we'll do some labeling. So here is my nuclear membrane starting to break down. This is my cell membrane. Okay, then over here, let's go ahead and draw a close-up of a chromosome. So this entire thing, you can call it a duplicated chromosome or a replicated chromosome. Both of those words mean the same thing. And I will use them interchangeably. So duplicated chromosome. Each half is identical. Remember? At the end of mitosis, we want 2 identical daughter cells. So each half is going to be identical, then eventually they will split and go into 2 different cells, then each cell can have exactly the same information, meaning that you can call one half of this a sister chromatid. Now I happen to circle the left side, but I also could have circled the right side. They are both called sister chromatids. And then this little center thing here is my centromere. When I'm counting the number of chromosomes in a cell, I'm counting the number of centromeres that are there. Okay, let's give yourself a second to write all of that down before we go onto the back and write what happens in prophase. Okay, so in prophase, the nuclear membrane or nuclear envelope disappears. This is when chromosomes are visible, meaning that my DNA has now condensed, wrapped around proteins, made itself easy to ship and take to different sides of the cell. Are visible. And last, I'm going to say this is the first step of mitosis. Okay, that's all we're putting on the back of this note card. Next is metaphase. Okay. So with metaphase, again, we're going to draw a cell. And this time, let's put a dotted line down the middle and label that cell equator. Now, when metaphase is happening, there's not an actual line that forms in the middle of the cell to say, here, this is the equator. It just is where all of the chromosomes line up during metaphase. It's always around the center. So scientists have just collectively called that the cell equator. Just like there's an equator on the Earth, but there's not a line all the way around it saying, hey, this is the equator of the Earth. It's just something that we all recognize. So this is the cell equator. And then in our last section, when we talked about organelles and what their different functions are, I told you that there was these noodly ones. At the time, all I said was, these aren't animal cells and they help with cell division. They help guide the chromosomes. Well, this is what I meant. The centrioles, you could think of them as like Spider-Man in your cell. How Spider-Man is able to shoot webs from his wrist and catch the bad guys, the centrioles are able to shoot what is called spindle fibers. And they attach here to the centromere, and then eventually these spindle fibers are going to shorten, and each is going to pull one half of this chromosome with it to the opposite side of the cell. So the centriole is like Spider-Man. You could also think of this as like a cowboy with his lasso, lassoing one half of this chromosome and pulling it to him. Okay. Here, a fun way to memorize metaphase, M for middle. The chromosomes line up in the middle of the cell. Okay, now let's go ahead and flip that over and write what's happening in metaphase. So in metaphase, let's center this. The chromosomes line up across the cell equator or middle of the cell. The spindle fibers attached to chromosomes and here the nucleus is gone. Notice we did not draw the nucleus. Okay, after this we are moving on to anaphase. Okay, all right, so for anaphase, still have our cell. Instead of being lined up across the cell equator, the different sides of the chromosomes, or the chromosomes are being pulled to opposite sides of the cell. Oh no, they've been pulled apart. So the centrioles still have the spindle fibers attached. The spindle fibers are shortening and pulling the chromatids to the correct parts of the cell. So let's label centrioles, spindle fibers, and this is a sister chromatid. And just how we mentioned in the Prophase note card, this is identical to this. So these are identical. These are identical, these are identical, these are identical. So then when this eventually splits in two, this side is identical to this side. Mitosis. Okay. So here, anaphase, we can think A for away. All right, and on the back, we're going to write what's happening in anaphase. Well, here, the chromosomes divide into two identical sets. Each half or set is pulled to opposite sides of the cell. We think A for away. And if you want to go back to your metaphase note card and add M for middle, you are totally free to do that. Okay, now we're almost done. We only have telophase and cytokinesis left. Okay. All right, telophase. Here's where the cell starts to look a little funky. It looks like this. Somebody said it kind of looks like a peanut. This is called a cleavage furrow. So this is where the cell is starting to pinch off here and it will eventually completely split. The nuclear membrane is starting to form back in each half. And now I've got my genetic material on both sides. They've been effectively split. They aren't